So I'm going to go ahead and introduce Dr. Cole for those of you that don't know him, which I'm sure most of us do. Dr. Cole is Assistant Professor, Division of Hematology and Oncology, Michigan State University, College of Human Medicine, Hermanos Cancer Institute, McLaren, Greater Lansing. Dr. Cole is the Director of Clinical Cancer Research, and his research interests include multiple myeloma and smoldering myeloma. His research interest includes multiple myeloma, non-Hodgkin's lymphoma, therapeutic apheresis, and myelodysplasia. Did I say that right?
Yes.
Okay, very, very good. So welcome, Dr. Cole. We're glad that you're with us this morning.
Oh, no, thanks. I'm always happy to wake up with you guys.
Oh.
Less complaining than with my daughters, who always seem to complain in the morning. So.
Okay. No, we're excited to be here with you. We're happy to be here.
Turn the camera a little bit more the other way, will you?
So can everyone? I'm trying to mute myself. Hi, Jackie.
Hi, how are you?
Good. How are you?
Great.
Good, good. So if anyone has any questions for Dr. Cole, we can go ahead and get started.
I have a question.
Okay, go ahead.
It's about COVID vaccines. I had my fourth Moderna COVID vaccine back in February, and I had Evusheld in March. And I'm wondering if you think I should have another COVID vaccine booster.
And let me just back up one step. Did you got the Evusheld? You said in March.
In March, yes.
And and one thing about the Evusheld that changed right around March, and I don't know if you talked to your doctor about this, is that the Evusheld is the pre-made antibody against COVID. And and so when patients have myeloma, we found out that the trick with vaccines is that vaccines are only as good as your body is able to make antibodies to the vaccine, and that patients with myeloma not only can, especially when they're on therapy, have lower response rates to the vaccine, but also they don't hang on to it as long. So the idea with Evusheld was that it is pre-made antibody to protect patients against COVID, which is guaranteed to work. It was released by the FDA, I think, in December or in January, but around March, the European FDA actually recommended a higher dose at 600 milligrams versus 300. And so around March, we started giving, instead of waiting 6 months to do the Evusheld booster, we were giving patients the additional 300. And so, one thing that I would, ask your doctor, or if is did you get the US dose, which was 300, or the now recommended dose of 600?
Well, I did have to get the second 2 shots because I only got the 300 originally.
Perfect. And you said that was in March. And so for the Evusheld, you're not going to be due probably until, I guess, September. And the good thing is that and when was your last booster? You said your COVID booster?
In February.
February, March, April, May, June. Yeah, so you'd probably be ready. Go ahead. You were going to say something. So you would probably, this would be a really good time to get that additional booster. And the reason is that it's been far enough away from the UV shield to allow your body to actually make antibodies. One thing that we found out is that if you give UV shield and the COVID shot back to back, is that the responses are low, of course, to the vaccine because you have tons of COVID antibody running around from the UV shield. And so we like to put, at least 3 or 4 months between UV shield and doing any of the COVID boosters. And so I would say that, it's been enough time where you could probably do that.
Thank you.
I'm the same as Hal. I had my 4th shot in February and my Evershell March or April. And then today I'm actually scheduled for my 5th shot at 1 o'clock today.
Perfect.
So any other questions for Dr. Cole? Well, Dr. Cole.
I have a question, Molly.
Okay.
If I were to go forward with the CAR T at some point, would the CAR T take away the lesions and the pain from the lesions that are in my ribs and my back?
So super good question. So, why do patients with myeloma have, bone disease? So why do you have pain there? It's twofold. One is that myeloma, unlike a lot of cancers, is very dependent upon the neighborhood that it lives in. And it only has really 2 or 3 partners that it works with. The primary partner that helps myeloma grow and proliferate are the bone marrow stromal cells, which is why myeloma is confined to the bone marrow compartment in the precursor diseases, the umbrellas and the smoldering myelomas. Why is it in the bone marrow? Because it has to live in that microenvironment with the bone marrow stromal cells. In the trick with the bone marrow is that there are other people living in the bone marrow, the red cells, white cell precursors, everybody else is in there. And space is at a premium, and myeloma then runs out of space to grow. So then it finds its next best partner, which are the osteoclasts that are in the bone, in the ribs and in the long bones. And the osteoclasts are the cells that balance with the osteoblasts, the cells that make bone, and you have the cells that destroy bone. So if I don't drink milk for days and days and days and days and days and days, months, my body will find calcium because those osteoclasts will just thin my bone a little bit and then release calcium. And then when I drink my milk and take my vitamin D, those osteoblasts will build up bone. And so there's always this constant yin and yang balance between the osteoclasts eating up bone and the osteoblasts making bone. The myeloma cells, like the osteoblasts, they're the next best thing to its favorite partner, the bone marrow stromal cells. And so the myeloma cells will then sit on those osteoclasts and then cause them to, they basically hijack them. They force those osteoclasts to give all the supportive food and cytokines that the myeloma needs to grow. And unfortunately, it thins the bones and then can cause bone fractures and the lytic lesions that we see. And so the primary way that we can treat myeloma pain is, of course, getting rid of those myeloma cells. And so will CAR T or any myeloma therapy that is effective eliminate the cells that are hijacking those osteoclasts? Absolutely. So part 1 of having the bones heal is solved, with the CAR T. Then what you have to do is that even, you could use CAR T to eliminate, and usually when people receive CAR T, their myeloma is eliminated very, very, very quickly. But does that change the pain? No, because only one of the three components to those lytic lesions have been addressed. So the second thing that needs to happen is that you need to get those osteoblasts that make bone to start working again. And so the drugs like Zometa, Xgeva, are all medications that decrease the osteoclastic activity that destroy bones and get those osteoblasts at going back to work. So will CAR T, help the pain? It'll help the pain, but the bone healing part then takes a bit longer to occur because you're going to need to have those osteoblasts start building bone and decrease the osteoclast activity, if that makes sense. And so when we treat myeloma, one thing that I tell my patients is that, you could actually have your pain get a little bit worse with treatment before it gets better. And the reason is that if the myeloma is in that component, is in that bone compartment, and it suddenly disappears, those osteoblasts haven't had an opportunity to kick back in. And now the bone's really a bit thinner because the myeloma that was in there is gone. Those osteoclasts are still chewing up the bone. And for a few, about a month or so, the bones can be a little bit weaker before they start getting strong again. And so it's just not, eliminating myeloma, but also, engaging those osteoblasts to make bone and decreasing the osteoclast activity, again, with the use of one of the bone strengthening agents, such as Zometa, Xgeva, also vitamin D, super important, and taking calcium. That, combination helps to build back the bones and decrease pain.
Nice metaphor.
Thanks.
Good.
So Dr. Cole, I have a question. So if there was one thing that you would like us all to walk away from this discussion, what would it be? So if all of your myeloma patients, what's one thing that you really want them to know?
Wow. What I think that when all of us are talking, all the myeloma doctors are talking, I would say that, and I think I've said this a number of times, but I think it's more true today than it has in the past, is how much hope that we have and how really excited everybody is. I've been doing this for on the high side of 15 years, well, a little bit more than that. And every year I've said, wow, things are getting better, things are getting better. But in the past, really 3 years, there has been a paradigm shift in myeloma therapy. I mean, things have really taken off, and like I've never ever seen before. There was a day when there were years between FDA approvals for myeloma. And when you had mentioned my introduction, actually me working with myelodysplasia and apheresis and all that other stuff, that is way back in the past. I don't do those things now so much. I primarily focus on myeloma. But the reason I was doing those other diseases is that there'd be years between anything happening in myeloma. And I needed to find another research interest because there was like, Revlimid, Velcade, and then years went by, and then there was carfilzomib, and then years and years and years went by. Then there was Pomalyst, and then years went by, and then everything else started happening. And now, it's what, last year we got 4 drugs approved by the FDA. In the past, 7 years, we've had 33 FDAs and drug indications have been approved, I think, in the past 7 or 8 years. I mean, it's unbelievable. And especially, so I think there's a lot of hope around what's going on with myeloma, more than I've ever seen. And I think the biggest thing I would take away is to definitely doing what you're doing right now and being, informed and empowered, because things are changing so quickly on the myeloma front, in every direction. What, 8 years ago, there was only one smoldering myeloma trial. And now I think last count is that I think they're like 30 smoldering myeloma trials, 40 smoldering myeloma trials. I mean, it is just, you have, Dr. Gobrel's PROMISE study, you have the ISTAP, Icelandic study, looking at precursor diseases. It's really unbelievable in what has happened. And really in the next few months, we're all have our fingers crossed that the flood of bispecific antibodies will become FDA approved. I think there's 6 bispecifics en route to the FDA as we speak. And when that type of novel therapy flood happens, that those benefits will span the entire breadth of myeloma. It will take literally 5 minutes before we start doing bispecifics and some of those therapies for smoldering upfront therapy for myeloma. I mean, it really is. It's going to be, in the next few years, are going to be an incredible few years, next 5 years in myeloma.
That's exciting.
That is exciting. Yeah, that's very good to hear. Yeah. So any updates on CAR T? Anything that we should know?
Yeah, actually there was a paper that was just published, I think it was Thursday, in Blood, about resistance to CAR T. And so, in brief, what CAR T is that they're re-engineered T cells, that we know that T cells are the cells that patrol our body against cancer cells. And so a T cell goes up to what should happen, that T cell should go up to a myeloma cell, and I'm going to have my wedding hand be the T cell, the good guy, and my right hand be the bad guy. And that T cell should engage the myeloma cell and kill it. What happens when patients have myeloma is that those T cells engage the myeloma cell. It asks, are you friend or foe? And the T cell says, must be a friend, and then moves on, and then the myeloma proliferates. And so what CAR T is that you can insert a gene inside that T cell so that it recognizes the myeloma cell as a bad guy and can destroy it. And which is really a remarkable therapy. The upshot about CAR T is that it's a one and done therapy, that you get one infusion of the T cells, of the engineered T cells, and the responses are, very quick. And patients, and it's a one and done, and there's really no other myeloma therapy other than, transplant that is just one time and done. And then you're not on any therapy. You have a complete holiday after CAR T. The other upshot about CAR T, the responses are phenomenal. That they're, on the upside of 80, 90%, and some studies at 100% pretty consistently. The downside about CAR T is that the responses are about maybe 2 years. And so patients, so it's not curative, and patients do relapse after they receive CAR T. And so now the hunt is on to see and the usual way that, or the usual theory behind why do patients relapse with CAR T is that we insert that gene inside the T cells for only one target. So the one target, BCMA, which is a protein that sits on myeloma cells, is that target for the T cell. So if this is BCMA on the myeloma cell, my T cell has been re-engineered, and all it's going to do is it's going to go for BCMA. So what the myeloma cells do, as you could probably guess, is that those myeloma cells mutate BCMA, so it's crooked, if that's a word. And then the T cells that used to engage BCMA just bounce right off, and they can't recognize it. And so so then the idea is that, well, is there anything else that's going on with resistance to CAR T? And one thing they found out is that there are other cells, as you can imagine, as we just already talked about, bone marrow fibroblasts that actually help to mask the myeloma cells from the CAR T cells, and that add to the resistance to using CAR T. So I think it was a group in Europe that looked at targeting these fibroblast cells that are helping myeloma cells avoid CAR T, and targeting those along with targeting BCMA. And what they found is that there are actually a couple interesting targets on those fibroblasts that actually can go after building back sensitivity to the myeloma cells to CAR T. So what they did, and I think this is so interesting, is that, and this is going to be where I'm very certain things are going to go, is bispecific CAR Ts. And what does that mean? Well, right now, like I mentioned, when we build a CAR T cell, we engineer it for only one protein, and that's usually BCMA. So what they're now doing is building bispecific CAR T cells. So it will not only go after BCMA, but go after another protein called SLAM7, or used to be called surface protein 1, but SLAM7. And so, and you now, instead of inserting just one gene inside the myeloma, inside the T cell, you insert two genes inside it, one for BCMA, one for SLAM7. And so now, and the bonus part is that those bone marrow fibroblasts also have SLAM7 on them. And so so now when you send in those T cells, it'll attack both SLAM7 and BCMA. And if the BCMA, I'm sorry. If the BCMA is broken, there's no way I can make this work without giving you the finger. So I gotta Gotta figure out another way. I'll use my pinky there. This is SLAM 7. This is BCMA. So if BCMA is broken, T cells go after SLAM 7. If you have both of them, T cells go after both of them, kill the myeloma cells. What if SLAM 7 mutates? Haven't seen that. If that mutates, T cells go after BCMA. Plus, those bone marrow fibroblasts that are helping the myeloma cell go on and helping the myeloma cell avoid the T cells are also knocked out by the bispecific CAR T. And so you're actually targeting not only the myeloma cell, not only 2 proteins on the myeloma cell, you're also targeting the cells that are helping myeloma avoid the therapy, be evasive to the therapy. And so I was like, that is super, super smart. And the upshot about that is that might be, that might be kind of the answer to how we can get myeloma patients to last longer with CAR T therapy. And one thing that, how do I explain this? So you've heard of allogeneic transplant and a donor transplant. So how do donor transplants work? So a donor transplant is really a primitive cellular therapy where you actually get some stranger's stem cells. And really what you want with that allo transplant is you want their T cells. Because if I got a transplant, if I had myeloma, my T cells aren't working, my sister's T cells will definitely kill my myeloma cells. It'll also hurt a lot of other cells in my body because it will realize it's not inside my sister anymore, but by me. And so my sister's T cells will build hundreds of cancer antigen receptors, tens of those chimeric receptor antigens on her T cells. And that's how allogeneic transplant works for myeloma, is that her T cells find all sorts of ways to kill my myeloma cell. Right now, we're in really the genesis of CAR T, where we do one cancer antigen at a time. My sister would have hundreds, but we do one at a time. When we start doing 2 at a time and 3 at a time, we're going to really build a cellular immune therapy where I'm not sure if the myeloma will be able to avoid it. Myeloma is pretty smart, but I think these cellular therapies and building this are really, I think, kind of the future. The downside about that is that they're more reactive right now. And so there's a little bit of worry about putting these bispecific CAR Ts in clinical trials because the cytokine release syndrome, when those T cells go kill the cells and release, a lot of inflammatory cytokines that cause, neurologic difficulty, the ICANS syndrome, and fever, capillary leak syndrome, and can sometimes cause heart failure and lung failure. We are really good at treating cytokine release syndrome with the current FDA-approved therapies for CAR T with only one, chimeric receptor antigen on it. The worry is that when you double that, you may double the cytokine release syndrome. And so we have to be kind of careful, but I bet you we're going to figure out how to do that. And then when we do that, that's going to be really, that's going to be something else. Does that sort of answer? So that, and I would also say, again, the bispecific antibodies, the great competitor against CAR T are the bispecifics that kind of work in a very similar fashion. And so, I think that, it's going to be really, it's going to be really interesting because, when the bispecifics get approved by the FDA, and there's so many of them coming down the pipeline, that's going to really put a very strong competitive pressure on CAR T to really up their ante, because that war between CAR T and bispecifics is just going to benefit researchers and patients because they have invested so much money into CAR T. And the bispecifics have invested so much money, they are going to fight to be the best on the market for sure. I'm done. Yes.
Dr. Cole, there's a couple
Oh.
of questions for you in the chat room.
Yeah, I think Mr. Brooks had a question. Then I'll do the things.
Dr. Cole, thank you. The FDA approval of these CAR T cell buy and try in the future therapies, is that going to be the be all we're all to get our medical coverages to pay for these kinds of very expensive treatments? Because I just lost someone just a week ago who lost their battle with multiple myeloma. And I think the issue, they didn't tell me directly, but I think the issue was with their medical coverage being able to cover them for CAR T cell. And I think that's what we're not discussing at this stage. And what can we do as a community of cure multiple myeloma to help the process?
Yeah, that's a super good question because you are 100% correct in that the efficacy keeps going up, but the cost keeps going up. And you're absolutely right in that, later today I have to write a letter to try to get the insurance company to call me because they don't want me, they don't want to, they're putting these barriers up to try to wear me out. So I have to write a letter to then call me to discuss a novel medication for one of my myeloma patients. I think the biggest thing, and I think people are very attuned to this in Michigan, is really putting their pressure on our congressmen and senators in the state level to really work at this. There is a sort of oral drug parity that's come out in Michigan. But the oral, and I'm very thankful for that. But the oral parity really does not address some of the new therapies that are coming out for myeloma. And we really do have to get, our the pressure on our congressmen and senators to really work at, helping to have the insurance companies and have really the healthcare system adapt to a lot of these changes. And so I would say that, in trying to really pressure them to get things going. And when I lived in Wisconsin, we did that. And we had, and I was part of that when I first got to Michigan, to really put pressure on them. And in Wisconsin, it worked. I mean, it worked really well. I was a bit surprised. And so I think the pressure on them could really help.
Thank you.
Yeah. So question, how does my disease, a plasmacytoma plus, differ in the osteoclast of, oh, that's a good question. So that's a really, so there is, some patients with myeloma will have, or some patients with plasma cell malignancies will have just a solitary plasmacytoma. So most patients, 80% of patients with myeloma will have some type of bone disease, Some patients will only have a plasmacytoma and have a few little plasma cells in their bone marrow. So not full myeloma, but very quiet myeloma with only, just the bone lesion. You can irradiate that bone lesion, and then the disease will be asleep for years and years, if not decades. How the interaction between the plasma cells and the osteoclast and osteoblast is very similar in solitary plasmacytomas as it is with myeloma. The one trick is that we think that there are other ways that the body is able to control the plasma cells from becoming full-blown myeloma. Some of that is the biology of those myeloma cells may not be aggressive and they're very slow at proliferating. And the other may be that the innate immune system is keeping pressure on those myeloma cells from expanding. But the general concept is very similar. Another question, my IgG went from 6,000 to 800 after starting therapy. Can you explain how the numbers work? So there are 3 tests that we use, 4 tests that we use to diagnose and to monitor myeloma. One is the M protein, the monoclonal protein. And for 80%, 75% of patients, that's the main test that we use, is that monoclonal protein, because the myeloma cells are the only producers of that protein. And so the higher that monoclonal protein, the more myeloma there is. The lower that monoclonal protein, the lower the amount of monomyeloma is. And so the main test look for when you have myeloma to see what's happening is what that monoclonal protein is doing. The other test is about 15% of myeloma patients will only produce light chains. The myeloma cells, instead of producing an intact antibody, will produce just fragments of it, and those light chains will be elevated. And so that's either the kappa light chain or the lambda light chain. And we use these days, we used to use 24-hour urines in order to measure that, but that was such a pain in the rear to collect your urine for 24 hours, and no one likes doing that. So now we just use the free light chain assay, which is a blood test. Again, the more myeloma you have, the higher that kappa or lambda protein. The lower the myeloma have, the lower that protein. The one trick about that protein is that it measures both normal and abnormal. So it's not as precise as the M protein, but it measures just what the light chain is. So it doesn't go all the way down to 0. It'll go down to the normal level for kappa lambda, which is about 2 milligrams. And for a lot of patients, it starts out at 100. And those are the 2 main tests that we check. The third one is the quantitative immunoglobulins. And so what does that mean? Well, the normal plasma cells in our body will produce IgG, IgM, and IgA. And you can measure those 3 normal antibody types in the blood using a test called a quantitative immunoglobulin. When patients have myeloma, they'll only produce 1. So if you have IgG myeloma, your IgG level, your M protein will be high, and your IgG level will be high. If you have IgA myeloma, you'll have a high IgA levels, and it will suppress the ones that are uninvolved. The other 2 that are uninvolved. That isn't as accurate as the M protein, because it measures both normal and abnormal. And so the IgG level will go down from 600,000 down to 800. But the M protein is the one that really is the best indicator of how the disease is going. So I hope that answered the question.
Yes. Yes.
the who and where are the most important myeloma lab research being done? Oh, I can't answer that question because I'm going to be partial to our lab. I'll just I know that our closest association is with Hermanos in Detroit, and we have a lab project. We have a lab here, a myeloma lab here at MSU, and we're super excited because our initial research has yielded some really interesting fruit that we're going to be presenting at the International Myeloma Workshop at the end of August. And so what our lab is looking at is that myeloma cells inside the microenvironment. Also, one observation that we've made is that when you do bone marrow biopsies of myeloma patients, you'll see that there's lots of fat in the marrow. And the reason that there's lots of fat in the marrow, the old idea was that, well, myeloma cells require a lot of energy, fat's where the energy is. And so there's going to be fat there in order to require the energy. Actually, it's much more dynamic than that there are adipocytes or the fat cells that actually help participate in myeloma growth. And we found a specific receptor that the myeloma cells use the fat cells in order to get energy from. So the energy transfer from the adipocytes to the myeloma cells, we've kind of found that connection that helps to feed them. And so our preliminary lab research in cell models, and we have a unique simulated microenvironment where we put adipocytes and myeloma cells in close conjunction with each other, and then we check the gene expression that happens between them, and that has some really interesting results that we're going to present in August. I think what's really interesting is that there are actually drugs, medications that can block the transfer of information and energy from those adipocytes to the myeloma cells. And so we've already talked to one of the pharmaceutical companies about a drug that they weren't sure what they were going to do with, that we may want to put into practice. Because one thing that we see is that patients that have obesity have worse outcomes with myeloma. And we really are interested is in myeloma precursor conditions, such as smoldering myeloma, because obese patients with smoldering myeloma have a higher risk of turning into myeloma. So what if we can block in those smoldering patients the transfer of information between the adipocytes and the myeloma cell? Will that help to arrest the disease without a lot of side effects that the current medications that we sometimes use for smoldering myeloma? Jeff Zonder's group at downtown Hermanos, they also have a lab in myeloma. And I think that's it. When I was at Michigan, we didn't have a myeloma lab, but we have one here, and I know Hermanos has one too. Oh, so next question. Dr. Colfer, someone had a UVA shield in June of 2022, booster in July. Was that too soon? No. Especially if the dose of the UVA shield was the lower dose, we do the booster to boost it up to the 600 total milligrams. And then that 600 is repeated every 3 months. Oh boy, what's the best maintenance therapy for dual diagnosis of myeloma and amyloidosis? Caroline Revelmid Darzlux, yeah. So the problem, one thing that we know when patients have myeloma and amyloid. So amyloid is when the myeloma cells produce that protein that I talked about, the light chains. And the light chains actually, instead of just floating around the blood, those light chains get folded into sheets and get inserted into the tissues, causing tissue damage. And so you get instead of just a one punch, you get a two punch, because you have the myeloma misbehaving, of course, and doing all the things that it does, but is also producing a protein which is causing tissue damage, especially to the heart. The receiving, one thing that is super exciting, that I think that everyone is excited about, and I don't know how well this has gotten out, but there really has been a major paradigm shift in how we treat newly diagnosed myeloma right now, by using now 4-drug therapy. And I think, most myeloma doctors that I talk to, a newly diagnosed patient is going to receive, and regardless of risk these days, is going to receive Darzalex or daratumumab, the antibody, plus the usual 3-drug cocktail. In every study that has been done where daratumumab was added to really any therapy, every one of those studies have shown, I specifically mean daratumumab, all those studies have shown benefit to the addition of standard therapy and adding daratumumab on. And so for maintenance therapy, adding on daratumumab is very good maintenance, super good maintenance therapy, in addition to Revlimid, especially if you had daratumumab as your upfront therapy. One thing that came out, which I think is very interesting, that we still need to try and get a better understanding of, is that there was the Cassiopeia trial. And the Cassiopeia trial was really interesting because it's a very European trial. It used a regimen that was Velcade, thalidomide, and I think melphalan or VDT. Velcade, thalidomide, and dexamethasone. And they had 2 arms. One was VDT plus Dara. The other was this VDT alone. So those two were put up against each other. Dara won that race. So case closed. After they had transplant, they then flipped the script. So the patients that received Dara up front, some of those patients were just on maintenance without Dara. And they did well. But the interesting thing is that the patients who were on VDT to begin with, and then only got the Dara in maintenance, also did well. So the idea of that is that maybe we keep thinking that we need to give everything up front and that maintenance is just, icing on the cake. But actually maintenance is very active myeloma therapy. And by adding on that Dara at the end gave the same benefits as having it at the beginning. And so, some patients that we have, especially the higher risk ones that didn't get Dara up front, are getting it in the maintenance therapy component of their therapy, based on what Cassiopeia has showed us. The question is that will some of not a lot of the myeloma trials that had that design, but I know that there are ones that are being worked on right now to further explore that. How important is a decrease in ion gap in addition to an increased chloride? So one thing that was, so there's a test called the anion gap, and it's a little math formula that is used to see what the blood, how many inflammatory proteins are in the blood. And so when patients have severe infections, that their anion gap can change. In the same way that, and it was recognized way back in the 60s, maybe 50s, that patients with myeloma have unusual anion gaps because of the monoclonal protein. And that you get rid of the monoclonal protein, the anion gap should then return to normal. So it's kind of a quick and dirty way by looking at the electrolytes to see what's going on with the myeloma. But definitely, there's so many things that can mess around with those lab values. It's definitely not as good as the monoclonal protein. Is it possible to have liquid? Is it possible to have liquid biopsies from regular blood or serum to find out myeloma markers with mass spec? There was just recently at the French meeting. I'm pretty sure it was the French. No, it was at ASCO. I'm sorry, it was at ASCO this year that they talked about circulating tumor DNA cells. And so myeloma cells, when they're reproducing or when they're dying from therapy, that they'll release mutated DNA into the bloodstream. And those mutated DNA are very specific for myeloma. And so there are ways to look at the blood to see what's going on with the myeloma. And the hope, the IMF has really been working on this, but other groups have been working on that maybe we don't need to do bone marrow biopsies anymore, but we can get some of the information from circulating tumor DNA. And the study that was presented at ASCO just kind of reaffirmed that you can do really very sophisticated testing on the peripheral blood looking at circulating tumor DNA. Because I would say that when we do a bone marrow biopsy, why do we do a bone marrow biopsy for myeloma? Well, one is to diagnose myeloma, but the diagnosis of myeloma really hasn't changed. Oh my goodness, in 122 years. 1900 was when Dr. J.H. Wright published a paper about doing bone marrow biopsies and myeloma. So we've been doing the same thing for 122 years to diagnose myeloma. Even though I think a lot of us can really figure out if someone has myeloma because they have bone lesions, anemia, all the other signs and symptoms. What is the bone marrow biopsy really giving you? It's really giving you what the cytogenetics have. So risk of myeloma is based on cytogenetics. And I would say that 90% of the time when we do bone marrow biopsies in newly diagnosed myeloma, I'm really hunting for the cytogenetics to see if they have high risk disease, standard risk disease, what mutations do they have. If we can get that same information from the peripheral blood by looking at circulating tumor DNA, that same prognostic information, then why are we doing a bone marrow biopsy? And so I think as this technology begins to progress, that I think we really could move towards moving away from bone marrow biopsies and going to liquid biopsies. In the same frame, you could also use it as a way to, because right now, if you want to measure if someone has minimum residual disease, you're going to have to do a bone marrow biopsy for MRD measurement. And so what everyone is really excited about is, can you do, again, the same thing by looking at circulating tumor DNA to see if someone has minimum residual disease. And so that is, something that's currently, I think, coming close to fruition. What has come to fruition is mass spec. And so right now, the SPEP that I just talked about, the main measure for myeloma, we've been doing that since the 1960s, the 1950s. That test is very, 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 very old. And mass spec is a way that can look at a much, much more sensitive way to look at monoclonal protein. And so, we actually send patients for their blood to Mayo for the mass spec test, especially if they have agaleel secretory disease. If they can just bring down the cost of mass spec, I think it will replace the M protein. It has to. It's way, way better. It's just the problem is that only a few places do it, and it's kind of on the pricey end, and the turnaround is way longer than it is with the M protein. You know, the M protein comes back in a couple days. Mass spec takes about a week to 2 weeks to come back. So if they can lower the cost and increase the efficiency, that'll be the test, because the mass spec can also be a very good surrogate for MRD measurement. And so, you know, you could get a twofer by being able to measure the M protein, diagnose the M protein using mass spec, and use the same test to help guide MRD measurement in the blood. I think that's all the questions. Are myeloma patients cured with remission MRD over 5 to, so the question is that if you're MRD negative for 2 to 5 years or 10 years, are you cured? And I would say the very specific answer to that is that one thing that we haven't quite done in myeloma is give a definition for what cured myeloma looks like. I think that this is a good point to end on. I think that we really do have the tools to potentially cure myeloma. We have to put those tools together. I mean, we have now a lot of tools on the, you know, in the shed. The trick is organizing those tools specifically for each patient to be able to put patients in long-term remissions, and in a long-term remission that lasts as long as your life is a cure. And so, but we haven't come up with a solid, reliable, universally accepted definition of what that looks like for myeloma. So I think in the next few years, I think that as we start to see already, you know, we're seeing remissions that are lasting, you know, decades. Hopefully we'll come up with a definition of cure that I can reliably convey to people to say, this is what we're hunting for. The same way that, you know, the VGPR, 90% reduction in myeloma, PR, 50% reduction in CR, same way we have those definitions, that we'll have better definitions for what cured looks like in the next few years. So, any other questions or any other thoughts? Oh, Molly, I think you're on mute.
There we go. Any other questions or comments?
Thank you, Dr. Cole. You're wonderful.
Oh, thanks.
Yes,
Thanks, Dr.
definitely.
Cole.
Yeah,
Thank you. Thanks for listening.
yeah, thank you, Dr. Cole. We appreciate your time.
Yep.
I love your visual analogies.
Oh, thank you. So, yeah, I'll make sure to tell my wife that she's a T cell. My right hand is my own cell. So,
Thank you.
Thank
Thank you.
yep. Take care.
Always a
everybody.
pleasure, Dr. Cole. Thank you.
Okay.
Yep.
Right.
Bye, everyone. Thank
Bye-bye.
you. Bye-bye.
Someday I'm gonna have to tell them I